My name is Thomas Brown. I'm the Executive Director of the Swedish Cancer Institute. I want to just very quickly once again recognize Linda and Beth, the co-chairs of this event, for their excellent work in the planning committee, but particularly to recognize each of you who are breast cancer survivors, especially those with metastatic disease. It's a very special event. I am moderating this session, and the title of the session is, of course, related to the clinical trial opportunity subject. It's developing a patient-centered research agenda. What I'd like to do, first of all, before I do that, I would ask Terry to introduce herself.
Hi. I'm Terry Plaster, and I'm a metastatic breast cancer patient. I've had metastatic disease for 13 years with mets to my liver. When I talk to you afterwards about that. And I have gradually and slowly gotten involved in advocacy over the years. I was busy taking care of my family at first and trying to stay alive, and then, as I like to say, my oncologist, Dr. Grelo, gave me a few nudges here and there to would I like to get involved in some different things, and I did, and I have, and I continue to get more and more involved in both patient and research advocacy. I belong to a group, Translational Breast Cancer Research Consortium. I'm the advocate for the University of Washington, and the first time I went, I didn't understand probably every other word they said. I went home, and I'm a little nerdle, so I made a list of all the acronyms, looked them all up, and put them in Excel so that I could quickly refer to them when they're talking about these things. And I'm constantly pulling on the sleeves of the docs at SCC. What does this mean, and how do you do that? And finally, I'm involved in some clinical trials, and I'm an advocate on several of those. I'm not afraid to tell people what I think might want to be included in things. And at different events, I'm not necessarily one that's going to stand up in front of a big group, but I will maybe go to the doctor or clinician afterwards and say, have you ever thought about this, or would you like to hear about that? Or, you know, insurance might be a problem for that. And that's how I.
So Terry, thank you for your leadership. What I would like to do before we get started with a series of questions that have been guided by our patient advocates, by many of you in the audience, is to make a few comments to sort of transition from the previous session relating to clinical trial opportunities to the concept of patient-centered research. I should explain, I'm a medical oncologist, and my research over the years has been focused on effectively early phase clinical trials, phase 1 work. And BK has alluded to that aspect of clinical trials. It's an area that has really changed, and that increasingly phase 1 work is heavily informed by preclinical data. He alluded to that. And the phase 1 trials tend to be more directed. Oftentimes we talk about phase 1/2 trials, because at the same time that we're trying to figure out the highest, safest dose and the side effect profile, we're also looking for real evidence of anti-tumor activity. The other role that I have is as PI on our personalized medicine research program. And certainly I think the era of genomics is beginning to inform the way we think about clinical trials. All of us here have been trained classically in the phase 1, phase 2, phase 3, phase 4 realm that BK alluded to. That to some degree is being turned on its head a little bit. People are now talking about the importance of big data, where you can accumulate large data sets on patient experience and mine that data almost as a research tool. Now, none of us feel that phase 1, phase 2, phase 3 trials will disappear, but I think we're going to see more research of the data mining type. So gathering big data, often from directly from patients, from people, is, as Corey has demonstrated, is increasingly going to to be an important endeavor. There's this concept of N of 1, and one has to be careful of that concept because there is virtue in looking at a large experience, but on an individual basis, increasingly we recognize that the organ site of origin is not always the most important piece of information. Increasingly, for patients with metastatic disease, it's the unique biology of your tumor, and you've heard that from various presenters today. And ultimately, the unique biology or fingerprint not only of your tumor, but of you. So that's now beginning to inform clinical trial conduct. And again, BK alluded to this concept of splitting and recognizing that if we see patients' tumors and we see human beings as unique biologic entities, then one can try to use that unique biological fingerprint to inform not only standard therapies but clinical trials. So the last three quick comments I wanted to make, and I think they'll inform the discussion. You've heard folks refer to testing on tissue during clinical trials and the importance of biological correlates. We know now mechanistically how many of these therapies work or should work or might work, and we try to look at the biological correlate. We try to see that indeed when we give the medicine, what we think is happening is truly happening. The other item, not only in a regional sense, but in a global sense, research is globalized more than ever. And some of the most exciting results in cancer research often come from another corner of the world. So it's important to have that global perspective. Clinical trials matching is part of the answer to the question that was asked about how one gets sort of a broad sense of what clinical trials are available. Cancer Commons is a terrific effort. Clinical trials matching is something that's very popular now in genomic medicine. We have an IT platform that is focused on taking the biological profile of the patient in terms of their tumor, taking their unique clinical characteristics from the electronic medical record, combining those two, and then running through a clinical trials matching algorithm that not only includes our clinical trials at the Swedish Cancer Institute, but clinical trials at SCCA or other institutions in the region. And I think increasingly you're going to see, certainly in the Pacific Northwest, a focus on that regional working across clinical barriers. And the last comment I would make, and in fact it's where I met Corey, we were reminiscing a little, I would comment on the moonshot, Vice President Biden's moonshot initiative, and both the Fred Hutch and by extension the SCCA as well as the Swedish Cancer Institute were participants in that event. And I think the main message from the moonshot was about the importance of big data and the importance of sharing information, especially research information, across traditional barriers. And of course, the objective of the moonshot is to achieve in 5 years in a research context what normally would take 10. That may not sound like a big deal, but it is. So I think the spirit of those assembled here is in that context. So with that, if you have questions, please send them up. But some homework was done before the session, and many of you had identified 3 areas that you wanted us to address. One was clinical trial design, the second was participation in clinical trials, and the third was transparency of clinical trial data. So what I'm going to do is start with clinical trial design, and BK, you had alluded to the broad nature of clinical trial design. There was a question about clinical trials often not addressing metastatic disease, and in particular, brain metastasis. Would you like to comment?
Sure. So I can start the ball off on this. I actually am privileged to be able to write trials. So I am a translational researcher. I literally see things that are going on in the laboratory, and when the light bulbs all go off, we say, is this something that's ready? Can we move it in? And so those are challenging trials to write. They're very scrutinized and so forth and so on. The brain mets are particularly a challenging space. Writing a trial for somebody with brain mets has to take into consideration not just the disease, but the patient, right? These are patients who oftentimes are very sick. You only got one brain, and most of us need it, unless you're in Congress. We have to consider the toxicity not only to the whole body, but to the
There
brain.
goes his federal funding.
Patty's got a brain. So it's challenging, and the other issue with a lot of these drugs is they don't get to the brain. We have wonderful drugs that clearly work below the neck, so to speak, but once they get inside the brain, they're either kicked out right away or just can't get to the target. So there's this thing called the blood-brain barrier, which we also have to address. So it's just a very challenging place to write trials. Having said that, it requires multidisciplinary effort. What we don't have represented here, I think, is Leah still in the room?
I think she has to go, but is Chris
Yeah,
still
so
here?
working with our other colleagues in the other disciplines, so the radiation therapists who address brain-based disease, as well as the neuro-oncologists who also help take care of this disease. We have to work with them as well. So it's just a lot of work.
I'm actually going to push back
Yes,
on
go ahead.
that just a teeny bit. So I was at ASCO this year, and I went to a session on clinical trial design, which was packed. I mean, it was, they actually had to have an overflow room because they had too many people who wanted to sit through it. And it was 3 presenters, as those sessions often are. It's usually 3 people talking about things. And so the first 2 presenters spoke, and then the third one was talking about how to include brain mets patients in your trial design. And half the room got up and left. And I just wanted to stand up and scream, like, sit back down. And I noticed, you know, I went to a separate session on brain metastases, and the way that the presenters spoke about those of us with brain mets was particularly disappointing to me. There was one of the presenters put up a slide that said, it had like a snowman and the doctor, and it was summer, and the doctor was like, I'm sorry, you only have 2 weeks to live, or something like that. And it's because, haha, brain mets patients die so fast. And it was awful. And so, and Leah actually and the gang in the brain mets session just were talking about how it's not actually the case that brain mets patients die as quickly as we thought they did. We used to not care much about cognitive function and how it's impacted by the treatment of brain metastases because the patients didn't live that long. But now we do. You know, I've been going for almost 1.5 years now with brain metastases. So, so I'm going to push back a little bit on that. So just put us back in the trials, man.
Well, one comment I would make, and I know it's true of the SCCA as well, is we both institutions have very dedicated neuro-oncology programs, and brain metastases are front and center as a research target. So I think the tide is turning, but I do recognize that there's truth in what
And I was going to say, I mean, I think the Hutch and Swedish are doing a great job. You guys are front, you know, sort of out there on it and pushing the envelope in a way that I think you may find if you're looking for trials at other centers, you may have more difficulty finding trials that you qualify for. So just something to be aware of.
And I completely agree with you. We have to take that stigma off of that. So if you have managed brain metastases, for sure, that's something that is a sticking point when we see trials being designed. And frankly, I'm not enamored with trials that just say excluded if have brain metastases. I think that's a cop-out. They're enriching their population. So we as investigators can be more aggressive with that too. Totally.
Right, you can argue that if there are valid reasons to consider those folks with brain metastases separately in an evaluation, but there's something called stratification that can be used for that purpose. It does mean you have to accrue more patients, but that should, one could argue that should more routinely be done.
Exactly, and that's how we find out if those drugs work on the brain. You know, they were talking in the brain met session again about there are drugs we thought didn't work on the brain, like TDM1, and it turns out they do. So if we include them, we find out.
So there's a related question regarding clinical trial design, and that is why don't pharma companies test drugs in metastatic models? I think one could argue the premise, but would some of you like to comment? Julie, would you like to comment?
So if the question is about
Not brain mets specifically, but metastatic disease in general.
Yeah, so if the question is the model in the lab, it actually is complicated. That we don't have many models and many cell lines, et cetera, to test this in the lab. We need we need better metastatic models. When we test metastatic breast cancer in the lab, we're frequently doing it in an incredibly artificial way. It's not where we have a cancer that arises in the breast in a mouse and then it on its own goes out and spreads. It's like we're forcing the spread in various ways by the way we inject the cells and all. It doesn't relate at all to what normally happens with the spread and progression. So we need actually, this is the importance of why we still have to fund basic science and all. We need better models for this where it is much more relatable to what happens in humans instead of creating these artificial scenarios that then when the concept doesn't work in a human, we then scratch our head and say why, and it's because it was a ridiculous model to start with.
So one of the interesting associated subjects that has been a focal point in breast cancer research is the concept of micrometastatic disease. Would you like to comment on that subject?
So, well, micrometastatic disease would be that you have distant disease, but you can't see it. I mean, that's when a person is diagnosed with what we think is an early stage disease, we make our best guess based on the features of the cancer and its stage and its grade and all of that on what do we think the chances that there already is micrometastatic spread. I mean, that's what we're trying to do in early stage disease when we estimate what's the chance of recurrence, what's the chance of death due to disease, and how can we decrease that with various therapies. We're treating micrometastatic disease in that setting, which means we just can't see it. And so it's that micrometastatic disease that you can get all the cancer in the breast through surgery and radiation, and you've left behind distant cells that you can't tell whether they're there. So we're working harder on how do we find distant disease, these liquid biopsies we talked about, using things in the blood. But the fascinating thing is, at the time of diagnosis, when you look hard enough with assays for things like circulating tumor cells and all, a lot of people do have evidence of cells that have circulated, and many of those, we've learned, will actually not recur. So there's something, it's different. Just being able to spread and get in the blood and get in the lymphatics is different than being able to survive in the bone or the liver or the lungs and then come back at a later time point. So lots of times we see circulating cells, but they don't have the genetic makeup that allows them to root themselves down and survive and then grow. So it's complicated because you can have micrometastatic disease that would never be able to actually come back and cause a problem, and then other micrometastatic disease that can, and we've got to do better at sorting out the difference between those, because otherwise we'd be giving everybody all the big guns therapy, and that's just too toxic.
That comment, Julie, also speaks to as we become more sophisticated and specific in our biological analyses, to include genomic analyses, it speaks to the importance of being sure that the data is interpreted properly, because it doesn't always mean what one might think it would mean, right? In the case of circulating tumor cells being present.
Right.
So Erin, would you comment a little bit as the director of our survivorship program at the SEI about research in the context of breast cancer survivorship and scientific wellness as a concept?
Okay. So what Dr. Brown is alluding to is a protocol that is being developed in conjunction with ISB, the Institutes for Systems Biology, trying to better understand some of the phenomena that happen to women post-chemotherapy. Many of you may have received adjuvant chemotherapy. I think it's a truism that most women have a period of unrelenting fatigue that lasts much longer than many expect. Often patients tell me that that fatigue has not really, those clouds have not parted until about a full year post-chemotherapy. We don't understand what's going on. So we're looking at inflammatory markers, we're looking at genomic changes, epigenetic changes, microbiome changes, and changes in the salivary hormones that may also correlate with some of these issues. So fatigue, arthralgias, potentially cognitive change. We're doing some neurocognitive testing in patients undergoing adjuvant chemotherapy, and some of the mood issues. So we're trying to better correlate what's been described in a very qualitative fashion with some hard science. That's basically what Dr. Brown is alluding to. The protocol's still in development. We're hoping to open it at the start of the year.
So I'd like to move on to ask Terry, you to comment a little bit about the aspect of transparency or the lack thereof when it comes to clinical trials, communication regarding clinical trials, your experience both personally and as seen through others.
Well, I think one of the things that's been trying is getting results back to patients, particularly if they're in a metastatic environment. Some patients may not be with us any longer, and how do we communicate that information to their families? Are they interested in knowing the results? And I think that just keeping the patient informed of what's going on is just a really big feat. And I know through the TBCRC, that's something we're always talking about. How do we get the results back to the patients? I just worked on a layperson summary, so I took the doctor's results from the clinical trial and I put them onto the website. And they're several years old. And I thought to myself, who even cares? This is so old now. If anyone even reads this, and we're spending so much time, the advocates, on this stuff. And I just think we need to get it back quicker. We need to do a better job of communicating it. And particularly when the results are not positive, I think that information needs to be shared. And we need to find a way, maybe some meta-analysis of results, so that when we do have exceptional responders to certain things, we need to be acting quicker on these things.
So this is on the NIH's radar. In fact, we have been handed some of these questions in advance of the panel discussion, so I did recognize that this was an issue that you were interested in discussing. I included this in one of the handouts. JAMA, one of the Viewpoint articles, dealt with the issue of trust and transparency in clinical trials, and it's written by Francis Collins from the NIH and Kathy Hudson. And I think this is front and center right now. I think there have been quite a few changes mandated by the NIH and FDA about how trials will be conducted in terms of quality, efficiency, data reporting, and they do a good job of outlining this in the article.
So Beth, would you comment from your perspective the general topic of transparency in the conduct of clinical research and communication with patients and their families?
When I try to read anything that comes out of any medical journal, I'm lost. And I'm a pretty highly educated person. I was an attorney before cancer took my career, so I'm pretty good at reading stuff and trying to figure it out. And it's not in English, right? It's in a completely different language, and so patients like Terry have to go through and try to translate it into something that we could possibly understand. That's the problem with the clinicaltrials.gov website, is that trying to find a trial on there and understanding, well, am I someone who is postmenopausal? I haven't had a period in 2 years, but before I had cancer, I had them regularly because I was 37 when I was diagnosed. So am I premenopausal? Am I postmenopausal? I don't know. What are all these drugs? How do I pronounce them? What does this mean? So, a lot of times they're not, it's very difficult for patients to access that information. And even, the other problem is that oftentimes it's published behind a paywall. So if I'm a patient and I want to know, I have a rare subtype of cancer called neuroendocrine breast cancer. There's about 20 US cases per year. So it's very rare, and mine doesn't even act like any of the ones in the literature, which I can't read because it's all behind a paywall. So I have to go to my oncologist and say, will you print this out for me? I saw this, I Google searched it, and there's this new study out, and so I think that is a serious problem with clinical trials and with research in general, is that for patients to be able to access it is still remaining extremely difficult. And as long as healthcare remains something that is a commodity instead of a right in this country, I think that we're going to continue to see that it's difficult for patients to control their information, to have access to the information they need to make informed choices, and to be able to get the treatments they need to stay alive.
So Beth, thank you for those comments. One of your comments about using the analogy of a different language points to another issue. One of the beauties of the United States is its diversity and its multiculturalism. And in clinical research, this represents the potential, unfortunately, for barriers, where individuals who might have a language other than English as their primary language, especially when one gets into genomically driven protocols, it's a morass, and sometimes it's even challenging to translate into another language.
You know, and it's not just a language issue. There are cultural things that prevent patients from participating in trials. Beyond the obvious issue of it can be very expensive to travel to a trial site. We're lucky here in the Northwest that we have such great researchers available, but say that you live in, Pasco, and you're going to have to come over here and live in order to participate in a trial. And how are you going to, so much for your job, and how are you going to pay your mortgage? And we know that patients who have cancer who go bankrupt are 50% more likely to die of their disease than if they do not go bankrupt. Those are actual lives that are being lost because of the cost of participating in trials and the cost of medications. So in addition, we have a history in this country of not treating our African American patients with respect, especially in the clinical trial setting. The Tuskegee experiments are one of the most egregious examples of mistreatment and experimentation on patients that exists in history. And so it's difficult for, if I come from a community where my ancestors have been experimented on by doctors against their will, I'm not necessarily going to want to enroll in a clinical trial. I'm not going to trust a doctor who says, I want to experiment on you, let alone if I don't speak English. It's hard enough for a patient who's well-educated, well-connected, has good insurance, is relatively healthy, isn't struggling with massive side effects from treatment, to find a trial. To then expect that of a patient who is being evicted because they, have lost all sense of income, all sources of income, that's why we're not seeing those patients accrue to those trials. And so I think we as a cancer community have to do a better job of reaching out to those communities so that we can include those patients in trials, and we can give them access to the cutting edge medicine that will keep them alive. Right now, if you're African American living in the United States, you have the same risk of dying of breast cancer as you did in 1975. And if you're white, you have a less risk of dying of breast cancer than you did in 1975, and that's wrong. All of the benefit in breast cancer mortality in the last 40 years has accrued to white people like me, and not to African American women. And I think that we can do better.
That underscoring your point that you and the panelists are well aware of is the concept that in modern day, one of the key features of high-quality modern-day cancer care is participation on clinical trials. That's essential to high-quality care. There's a question here that I think is an interesting one, and that is, do trials take into consideration age at diagnosis? And from the comments made here, I think in particular older age, but maybe also a very young age. Would any of the panelists like to comment?
So I don't know of examples of metastatic trials that would take into account the age you were originally diagnosed, because some people will relapse 2 years later, some 15, 20 years later. I don't know of any precedent for that. And then the question is, do trials take into account the age at your metastatic diagnosis, which might be more of the question. And we certainly, with our NIH and CI sponsored trials, have really tried to take age out of the picture and do it more based on what your performance status is and all. We used to have a lot of trials that you couldn't be more than 70 or something. But we've tried to take that out, at least of our government trials now. Certainly trials focused on younger women who still have functioning ovaries would be an example of where we do take into account the age, because if you're still premenopausal, you have to be pretty young. But I don't know. But
and I just add that what performance status means is, just to define that, is basically how healthy you are. Are you healthy enough to be, trying an experimental medication?
So I wanted to slip one more question in, a card that came down earlier, and it does connect to a previous subject, but it more importantly connects to the topic, Terry, of communication. So I'd like maybe for you to be the first person to comment. It says, when will the doctors more clearly communicate to those early stagers that the likelihood of the micrometastatic disease will return more likely than not? It impacts the decisions, especially for younger women. And the final comment is, doctors sort of, quote unquote, dance around that issue, leaving you wondering. And for me, this comment question is more a comment on communication. So Terry, would you?
I'm really not sure how to answer that. I think that there's a very fine line there between too much information that can make people worried and not enough information so that people are empowered. And so I just feel very hazy on this. I don't have a really strong opinion.
Strong opinions. Is anyone shocked? No. I wasn't someone who was an early stager who later metastasized, so I'll put that out there right there. I was stage 4 from diagnosis. So I have a little bit different perspective on things than somebody who, one of the pictures we have out in the hallway is Holly Kitchen, who was part of the project that Pfizer did, the Story Half Told project, and who passed away last January. And she did a video of herself that went viral where she talked about how she thought she was cured. And they used the word cured, and they told her she was cured, she had nothing more to worry about, and she's dead now. And she didn't know that this was a possibility. And so when it hit her, it was even harder, I think, emotionally, than for someone like me who knew right away, right? So I didn't live in this false hope. That said, I think it's also important, I talk to my early stage friends and they worry about every single ache and pain that comes along,
Yes.
which is also not useful. So I think you're right that there needs to be a balance between, I think it's important for doctors to say it's possible that your cancer could come back someday, but we really hope that it didn't. And we really hope, we've done everything we can to make that so that you will not have to have cancer again. But I think that language is really important, and you have to be careful.
I totally agree with that. I also was, as my husband and I say, we used the C word, which was cured. They told me I was cured, so I was shocked when my cancer returned. But like Beth said, I just think it's really important that, you just have a good rapport with your oncologist or, any of your doctors. You feel comfortable asking questions and that you make your relationship with them what you want it to be and what works between you and them for knowledge and information.
If you don't trust your doctor, you have the wrong doctor. Get a new one. There's better ones out there.
On that important note, I want to thank each of our panelists, especially Terry and Beth, and thank you to the audience.